Now about other vectors. First is come to cosmid vector. Cosmid vector. As the name suggests, for the first time, J. Collins and B. Horn first developed the cosmid in 1978. So, as the name suggests, it is a vector that are constructed by unite some part of plasmid, that is plasmid DNA, plus cos site of phage lambda. So plasmid plus cos site of phage lambda, which becomes cosmid. Means this is the vector which is constructed by using plasmid as well as cos site of phage lambda. They have origin of replication, the regions such as origin of replication, selectable marker gene for antibiotic resistant, and other recognition site for other enzymes like in other vectors, and side by side cos site, which is also known as cohesive site. So because of this cos site, this vector gets circularized through cos site. The cos site is present to circularize this vector, and it can replicate as a plasmid. The upper limit of foreign DNA to be packed into plasmid is 45 kb. Means this is the upper limit which can be packed in this plasmid. So finally, a cosmid vector has a cos site, ORI region, ampicillin resistant gene, as well as restriction sites for various enzymes, and 45 kb up to 45 kilobase foreign DNA can be packed into this cosmid. Come to the next vector known as YAC. YAC vectors. YAC means yeast artificial chromosome. Yeast artificial chromosome. Now what are these YAC vectors? Its full form is yeast artificial chromosome. They have been developed as high capacity vectors which can clone very large DNA segment about 1 MB in length. So if we want to insert a large DNA sequence, a large DNA segment, then we can use these YAC vectors. These vectors were most commonly used. It has been used for physical mapping of human chromosome in Human Genome Project. So at that particular time, these vectors were developed so that they can be cloned in a large chromosome. These are high capacity vectors. These yeast vectors contains following sequences. It is maintained as a separate chromosome in the cell, and as I told you that they are used for physical mapping of human chromosomes in Human Genome Project. Their plasmid contains this yeast vector. It contains E. coli origin of replication, means ORI region for E. coli. It contains E. coli origin of replication. These are about YAC vectors. So it has E. coli region for replication and selectable marker, basically ampicillin resistant gene. Normally this ampicillin resistant gene is present in this YAC vector, and in addition to all these genes, it has a yeast DNA sequence, genes for uracil biosynthesis pathway. They have gene for uracil biosynthesis pathway, URA. This is uracil biosynthesis pathway, and uracil is an amine. And after that, the uracil biosynthesis pathway, there are the genes which provide centromeric function. CEN genes. There are different genes. They provide centromeric function. Autonomic replication sequences, ARS. This region is also present in these yeast artificial chromosome. Genes for tryptophan synthesis. Tryptophan is an amino acid. The genes for tryptophan synthesis are also present in this particular vector. They have the necessary sequences from an E. coli plasmid also. As I told you that telomeric sequence in yeast chromosome is also present. It has necessary sequences for an E. coli plasmid. Selectable marker genes are present. LEU2 gene is also present, which codes for leucine. So all these genes are present in YAC vector. Now one more important vector, that is BAC vector. BAC is bacterial artificial chromosome. Bacterial artificial chromosome, these are used as cloning vectors. They are constructed by using fertility factor of E. coli. the factor, fertility factor, which is present in E. coli. So they are based on that fertility factor. Back vectors contain ORI gene, means the region for origin of replication, a gene for maintenance of F factor, selectable marker, means an antibiotic resistant gene is present. Like other vectors, it also has antibiotic resistant gene. It has many restriction sites for insertion of foreign DNA, and the upper limit of foreign DNA to be inserted in back is about 300 to 3500 kb. The upper limit is 300 to 3500 kb. So these are back vectors. So the genes which are present in back vectors, again I am telling you, it has ORI region and antibiotic resistant genes for different antibiotics, and maintenance gene for maintenance of F factor, restriction sites for many enzymes, so that generally multiple cloning sites for many enzymes, so that we can use different enzymes for the restriction of DNA, and upper limit is 300 to 3500 kb. So these back vectors were also used during genome sequencing projects. So it is used in genome sequencing project just as yeast or yak vector. Last, just we'll discuss about animal viral vectors as well as plant viral vectors. So in nature there are several viruses which cause disease. These virus gets absorbed to the body surface of suitable host and then infect the cell. All of us know about this. So this ability of animal viruses has been exploited, and virus-based vectors have been designed so that foreign DNA can be introduced. Foreign DNA with a known function can be introduced into cultured eukaryotic cells. For this purpose, there are many viruses which can be used, such as simian virus 40, that was SV40, is one of the animal viral vector based animal viral based cloning vector, and it was constructed and used in cloning experiment using mammalian cells. That was the first time SV40 based cloning vector was constructed. Since then there are many vectors which were constructed. They are adenovirus, papillomavirus, retrovirus, baculovirus in insect cells can be used, and this papillovirus and retrovirus are used to clone foreign DNA into the mammalian cells. So these are animal viral vectors. After animal viral vectors, now the last is about plant viral vectors. Now what are these plant viral vectors? As we know that there are several plant viruses which can cause serious loss to the crop plants. Some of the virus such as tobacco mosaic virus, cauliflower mosaic virus, they have been used as vectors, and they have success have been achieved to a limited extent. So just if we want to make any transgenic plant and we want to introduce our gene of interest in any of the plant, so we have one option to use plant viral vectors. So these are the plant viral vectors which can be used as a vector in some plants. So that's all with the vectors. In our next session, we'll discuss about host cells.